This presentation is about pathophysiology of cancer. We can define cancer or characterize cancer in terms of being benign or malignant. And how we do this is understanding the growth, the capsule characteristic, whether it's invasive or not, the differentiation, mitotic index, and the metastasized, is it metastasized or is it not metastasized? So when I think in terms of growth, cancer can grow very fast, ranging in different speeds, or it can be very slow as well. And some cancers may elicit slow growth and others are very rapid. In terms of the capsule, the presence or absence of the capsule around the tumor can be an important characteristic. A well-defined capsule indicates that the tumor is encapsulated and contained within that distinct boundary. But on the other hand, if the tumor is not encapsulated, it doesn't have a well-defined boundary, and so we worry about the spread of that cancer. Whether the cancer is invasive or not, this is telling us that if it's invasive, it has the ability to invade the surrounding tissues or spread to distant sites, versus a non-invasive cancer, also known as carcinoma in situ, remains localized and it doesn't invade any nearby tissues. Differentiation refers to how closely the cancer cell resembles a normal cell, or the cell in which it originated from. Well-differentiated cancer cells resemble normal cells, both in structure and function, but poorly differentiated cancer cells, on the other hand, have a more abnormal appearance and function. The mitotic index, high or low, this measures the activity of cancer cells, the activity of dividing. It indicates the rate of the cell division within the tumor. So if a cancer has a low mitotic index, this suggests that the tumor cells are dividing at a slower rate than a high mitotic index, which is a rapid cell division. Metastasis is the spread of cancer, and this is the spread from the original site to other body parts. And the presence or absence of metastasis is an important characteristic of cancer. If it's not metastasized, it remains confined to the original location. However, if metastasis has occurred, cancer cells have already now spread to distant organs and different tissues. The characteristics collectively provide valuable information about the behavior of the cancer, the aggressiveness of the cancer, and they help the healthcare professional in making the diagnosis, diagnostic and treatment decisions, and it also helps to assess the prognosis of the patient. So I just want to further define carcinoma in situ. So this is the pre-invasive epithelial malignant tumors, but they're confined to the original site of the origin, and they haven't broken through the basement membrane or invaded the surrounding stoma. The epithelial origin, or when we talk about in terms of carcinoma in situ, it is granular or epithelial tissue, and they're found in various organs and structures throughout the body. Remember, epithelial tissues line the surface and the cavities of organs, and the glandular tissues are specialized for secretion. Carcinoma in situ, or CIS, is considered a stage of cancer where abnormal cells have acquired malignant characteristics. So they haven't invaded those deeper tissues and they haven't spread to the other parts of the body. The tumor is localized, confined within that epithelial layer. And this is the pre-invasive nature of the cancer. They haven't breached the basement membrane, they haven't moved out into other areas, and this acts as a barrier between the epithelium and the underlying tissues. Because the CIS tumor has not invaded the surrounding stroma, which refers to the connective tissue and the supporting structures around that epithelial layer, the absence of this invasion in the stroma distinguishes CIS from other invasive cancers. where in those cases the tumor cells have breached the basement membrane and invaded the other tissues around it. Carcinoma in situ is significant because it represents a stage where early detection and intervention can be highly effective. Identifying and treating carcinoma in situ can help prevent the progression to invasive cancer. It provides an opportunity for timely treatment to remove any abnormal cells and to prevent the development of invasive tumors. So why is this significant? Well, in terms of the histopathological examination of tissue samples that we can get through biopsies and other diagnostic procedures, we are able to see this characteristic on a microscopic evaluation, obviously, of the tumor cell. And we can look at the location relative to the basement membrane and the absence of the stromal invasion. The management of carcinoma in situ depends on the specific type and the location of the tumor, and so does the treatment options, which we have talked about already. Carcinomas arise from epithelial tissue. Leukemias are blood-forming cells. Carcinoma in situ refers to the pre-invasive epithelial tissues of glandular or squamous cell origins. So I want you to know these three early stages, or the early stages of cancer, the localized to the epithelium, and know that they haven't penetrated that local basement membrane. I want you to understand this as we move forward so that you should be able to talk about it and differentiating the types of blood-forming cancers versus carcinoma in situ and other carcinomas that have spread or become malignant. Okay, so let's talk about the transformation of a normal cell. Normal cells require specific growth factors, and these growth factors are going to stimulate proliferation and growth. But in contrast, cancer cells have acquired some alterations in the way these pathways and the signaling pathways are enabled, and they can bypass the usual dependency of growth factors. And so they proliferate and multiply even when there are no external growth factors present, and this leads to uncontrolled cell growth. With a normal cell, they require these external growth factors to actually grow. When we talk about contact inhibition, this is the mechanism by which normal cells stop dividing and growing when they come in contact with a neighboring cell. Cancer cells, however, they don't have this ability to continue to divide and grow uncontrollably, even when they're in close proximity to other cells. This lack of contact inhibition contributes to the formation of tumor masses and their potential to invade the surrounding tissues. They don't know when to stop. Normal cells typically require attachment to some type of solid surface or extracellular matrix to grow and divide. But cancer cells, they can acquire the ability to grow and multiply in the absence of this anchorage to a solid surface. So that's what it talks about, anchorage independence. They can detach from their original location and survive and proliferate on their own, suspended or floating. And this anchorage independence allows cancer cells to invade and metastasize to different sites. So a normal cell would be dependent and have to anchor itself to a solid surface. Normal cells have a limited lifespan. They undergo the process called senescence. And this is when they know to stop dividing after a certain number of cell divisions. But in contrast, cancer cells don't have that ability to bypass this, and they become essentially immortal. And they continue dividing and dividing and dividing indefinitely without the usual constraints on cell lifespan. So the immortality is partially achieved through the activation of telomerase, which is an enzyme that maintains the length of telomeres, which are the protective caps on the chromosomes. So all in all, these characteristics collectively contribute to aggressive, uncontrolled growth of cancer cells. They enable them to proliferate without the usual regulatory mechanisms that govern normal cell growth, and they can just invade. Tumor suppressor genes, they're also called and often referred to as anti-oncogenes, are a class of genes that play a crucial role in protecting cells from developing cancer. They regulate cell growth and prevent the formation of tumors and maintain a genomic stability. The tumor suppressor genes, they are what protects a cell from inhibiting cell division. They promote repair of the DNA and apoptosis, which is cell death, which is necessary. They also help maintain normal cell function and prevent the accumulation of many genetic alterations that can lead to uncontrolled cell growth and the development of cancer. When a tumor suppressor gene undergoes mutation, however, it results in a loss of reduction in its normal function. The mutation can occur spontaneously, or it can be caused by external factors such as an exposure to carcinogen or inherited genetic abnormalities. The last thing I want to say about mutation loss of function is that a tumor suppressor gene can remove the so-called brakes on the cell growth control, which allows abnormal cells to just multiply and multiply and multiply. In most cases, a mutation of the tumor suppressor gene, it's not sufficient to cause cancer, but significantly increases the risk of cancer development. And this is the combinational effect. So cancer typically arises due to the accumulation of multiple genetic changes, including mutations in other tumor suppressor genes and oncogenes. Remember, the genes that promote cell growth. The loss of the function of the tumor suppressor gene is often a critical event in combination with other genetic alterations that are occurring, and this drives the progression of the cells to a more cancerous state. Proto-oncogenes, these are normal genes that present in cells that have the potential to transform into oncogenes through alterations or mutations. Oncogenes are genes that promote uncontrolled cell growth and contribute to the development of cancer. So make sure you know the difference between the two and what happens with proto-oncogenes. Proto-oncogenes are important in the normal cellular processes, and they regulate cellular activities. They play a role in maintaining balance between cell growth, differentiation, and apoptosis. Again, that's programmed cell death. One of the key functions of proto-oncogenes is to provide signals that promote cell division, and these genes help regulate the cell cycle. They ensure the cells divide when needed for growth, repair, and development. They activate signaling pathways that stimulate cell division, and this allows cells to replicate and proliferate. Proto-oncogenes also play a role in regulating apoptosis, like I said, and the process of the programmed cell death is needed. They are involved in controlling the balance between cell survival and cell death. When the functioning occurs properly, the proto-oncogene will ensure that cells undergo apoptosis when it's necessary, and then it will eliminate the damager in unwanted cells. This helps to maintain healthy tissue homeostasis, and it prevents the survival of cells with potentially harmful genetic alterations. So it knows when to get rid of things that should not be there or can present as a potential threat. But when a proto-oncogene undergoes a mutation or alteration, its normal regulatory functions can be disrupted. The altered gene can become an oncogene at that point, which drives excessive cell growth and division and leads to uncontrolled proliferation. It reduces the ability for apoptosis and the formation of tumors. So we have to understand the proto-oncogenes and how they're essential for cancer research and treatment. And if we can identify specific proto-oncogenes, and the pathways that regulate them, then we can give some insight on underlying cancer development. Therefore, more targeted therapies can help block and inhibit the activity of those specific oncogenes. An oncogene is a gene that has the potential to cause cancer. So just with all of that technical language that we just went over, I just want to make sure that you understand that it's not that an oncogene is cancerous, but it does have the potential to become cancerous. So you think of when we test patients for specific cancerous genes, we're looking for this oncogene to say we could produce cancer, but it doesn't mean that if they are positive for this certain type of gene that they have the cancer then. But this helps people to sometimes use a more prophylactic approach or upfront approach. Think about women that are testing positive for the BRCA gene and they feel like, well, I think maybe this has a potential to cause cancer, that I might want to intervene before there is cancer. Identifying specific oncogenes and detecting their presence or alterations in tumors can help provide valuable information for diagnosis, prognosis, and personalized treatment decisions in the future. When we talk about point mutation, this is the alteration in the DNA sequence of a gene. And this involves the substitution, or it could be the deletion or an insertion of a single nucleotide base that should not be there. And in the contents of oncogenes, point mutations can lead to the activation or overexpression of the gene, and this can develop or contribute to the development of cancer. When we talk about translocation, this is the genetic abnormality in where part of the chromosome breaks off and it attaches to another non-homologous chromosome. So two things are happening here and they're translocating themselves where they should not be. This rearrangement can result in the fusion of two different genes, including oncogenes, leading to the formation of a hybrid gene called a fusion gene. And translocations can also involve oncogenes that have been implicated in various types of cancers like Burkitt's lymphoma and Hodgkin's lymphoma. With Burkitt's lymphoma, the translocation involves the CMYC oncogene. This is very characteristic of Burkitt's lymphoma. This is a type of non-Hodgkin's lymphoma. The translocation with the CMYC gene usually happens from chromosome 8 and the immunoglobulin gene region on chromosome 14. The fusion events to the overexpression of the CMYC protein, and this promotes uncontrolled cell proliferation. With chronic myeloid leukemia, we can see specific translocation on the Philadelphia chromosome. This is important to understand the Philadelphia chromosome. It involves the fusion of the BCR, which stands for a breakpoint cluster region, on the gene on chromosome 22, and the ABL1 gene on chromosome 9. So just know it's 9 and 22 for chronic myeloid lymphoma. And so when this happens, we can have uncontrolled cell growth with chronic myeloid leukemia. We also want to understand gene amplification, and this refers to abnormal duplication of a particle of a gene or a genomic region. And in cancer, gene amplification can lead to an overexpression of oncogenes, and this increased activity promotes tumor growth. Let me give you an example. If we have an amplification of an oncogene that's been observed in a certain cancer type, for instance, the HER2, human epidermal growth factor receptor 2, HER2. This is commonly found in breast cancer. So if we have the HER2 amplification and we have the increased expression of the HER2 protein, this could lead to uncontrolled cell growth and aggressive tumor behavior. And if we see the specific genetic alteration in this oncogene, such as point mutation, or we see translocation or gene amplification, then this is going to give us valuable insight on maybe the mechanism that's going to drive cancer development in the future. And this picture shows you an example of the Philadelphia chromosome, the translocation of chromosome 9 and 22, which signifies or is prominent in chronic myeloid leukemia. And then this was the example for the MYC gene, where it's chromosome 8 and 14 that gets transformed, and you can see this in Burkitt's lymphoma, which again, this is a non-Hodgkin type lymphoma. Okay, so when the cell undergoes mutation or inactivation, the normal tumor suppressing function becomes disrupted, and it leads to unregulated cellular growth and an increased risk of cancer development. What's the role of a tumor suppressor gene? This tumor suppressor gene is supposed to be like the brake on cell growth and division, and it's responsible for inhibiting any more progression or proliferation of the cell cycle. It promotes DNA repair, and it also promotes apoptosis when it's necessary. Mutations in the tumor suppressor gene can occur through various mechanisms, including point mutation, which we saw, chromosomal deletions, insertions, or any type of epigenetic modification that happens. And when we have these genetic alterations, they can result in a loss or reduction of function of the tumor suppressor gene. They can lose cellular control when the tumor suppressor genes are mutated or inactivated. So cells lose their ability to respond to the signals that would have normally said, hey, don't proliferate. And then now we have unregulated cell growth and proliferation, and we have more accumulation of genetic alterations. It puts the person at increased risk of developing cancer when we have inactivation of the tumor suppressor gene as well. So examples of the tumor suppressor genes are the BRCA1 and 2. Also could be signs for certain types of cancer, such as breast cancer, APC, PTEN, examples for ovarian, colorectal, and prostate cancer. So not too important at this point what type of, or the specific tumor suppressor genes, but just knowing that tumor suppressor genes are very important in the identification of cancer and the inactivation in cancer development. The genomic stability of a cell is essential for maintaining the normal function and preventing cancer. However, there are certain factors that can increase the tendency for these genomic mutations to occur. Throughout the life cycle of a cell, including replication of DNA, repair, cell growth, and other cellular processes, the processes are susceptible to errors. And when they lead to genomic mutations, it's because there are some errors in this life cycle of the cell. We also have factors such as exposures to environmental carcinogens like secondhand tobacco smoke, radiation. This is going to increase replication of more DNA errors. Or we might have reactive oxygen species, endogenous DNA damaging agents, and deficiencies in the repair of DNA itself. Genomic mutations have the potential to disrupt the normal cellular functions, and this can also lead to the development of cancer. So accumulation of mutations in critical genes, including oncogenes and tumor suppressor genes, can drive uncontrolled cell growth and the formation of cancer. So what is a caretaker gene? Caretaker genes play a big role in maintaining this genomic stability because it encodes, or it's an encoding protein in DNA repair. These proteins can detect and correct errors for damage in the DNA sequence, preventing the accumulation of mutations. So caretaker genes are important. Let me give you an example of a DNA repair protein encoded by a caretaker or caretaker gene. If you think about excision repair, a nucleotide excision repair, a mismatch repair, a double-stranded break in the DNA, that type of repair, those all need to happen as examples of DNA repair proteins. If you have a loss of caretaker gene function, or the mutation or inactivation of a caretaker gene occurs, this can impair the DNA repair process altogether. So we need caretaker genes to function correctly. Genomic instability can result from increased silencing or modulation of that gene function, and the promoter regions of that gene may be altered. And so because of this alteration, the gene becomes silenced, and we have an altered gene expression. What's a promoter region? These are crucial regulatory regions of that gene that control the transcription and the expression. And once they are altered, these promoter genes can disrupt the normal gene regulation. But altered promoter regions lead to the silencing, and so the tumor suppressor genes are silenced, and the activation of oncogenes will occur and promote uncontrolled cell growth and tumor formation. In terms of chromosome instability, this is a common feature actually of a cancer cell, and it refers to abnormalities in the structure and the number of the chromosome. And it results in defects of DNA repair and abnormalities in the cell cycle itself. How is it manifested? As a whole, parts of chromosome as well. It can also be a deletion of a tumor suppressor gene or any other critical genetic material. The loss of genetic material can contribute to uncontrolled cell growth and cancer progression. Telomeres are specialized structures located at the ends of the chromosomes. They play a crucial role in the cell division and the lifespan of the cell. Going back to the transcription, we talked about telomeres there, but telomeres, they act as those protective caps, preventing the loss of the genetic material, and it maintains the stability. In normal body cells, each division brings the telomere closer to the critical limit, and as telomeres become shorter with each cycle of cell division, they eventually reach a threshold and triggers cellular senescence, which is apoptosis, programmed cell death. The limited ability of a cell to divide is known as the Hayflick limit, Hayflick. Telomeres, or telomerase, is an enzyme that can actually elongate telomeres by adding repetitive DNA sequences to the ends of the chromosomes, and this helps to maintain the length and integrity of the telomeres. In normal body cells, this telomerase activity is typically low or absent, leading to telomere shortening over time. The shortening is associated with cellular aging and age-related diseases. But when telomeres become critically short, the cells lose their ability to divide and function properly altogether. And this can lead to tissue dysfunction and more age-related conditions. One hallmark of a cancer cell is the ability to bypass that Hayflick limit. And they can achieve immortality by activating more telomerase, and so they don't understand that they are not supposed to grow anymore or proliferate, and they will continue. Cancer cells can upregulate telomerase expression, allowing them to maintain their length and sustain cell growth, and so they continue to proliferate, and this leads to tumor growth and progression. Angiogenesis is a term that refers to new blood vessels being formed from pre-existing blood vessels. So not only is this cancer cell proliferating, we also have new blood vessels feeding this new tissue. Angiogenesis plays a critical role in tumor growth, invasion, and metastasis by supplying oxygen and nutrients to that growing tumor mass. And so the tumor continues to grow, it's going to require more blood supply. But with angiogenesis, it allows it to grow because it's sustaining the growth and metabolism by this increased blood supply. Advanced cancers have the ability to secrete angiogenic factors. And what that means is that they are able to stimulate the growth and migration of endothelial cells, and they will continue to build more blood vessels because endothelial cells are the building blocks of blood vessels. So as they promote the formation of new blood vessels, it again induces more proliferation, differentiation of endothelial cells. The angiogenic factors that we're talking about are the vascular endothelial growth factors, or the VEGF. Okay, we talked about apoptosis a lot, but I want to just define it here in terms of cancer. Programmed cell death, it's crucial in physiological processes because they help maintain tissue homeostasis. And when we have a damaged cell or an unwanted element, it eliminates it. But in cancer, there's defects in that apoptotic pathway, and it contributes to the tumor's development, progression, and resistance to cell death. Apoptosis itself, this is a tightly regulated process, and there are a series of cellular events that lead to controlled cell death. The activation of signaling pathways, it dismantles the cell. There's phagocytosis of the apoptotic cell and the debris. But when apoptosis doesn't happen, the cell will continue to proliferate, or the damaged cell will continue to exist. Apoptosis can be initiated through intrinsic pathways and extrinsic and intrinsic pathways. The intrinsic pathway is activated by the extracellular signals such as DNA damage or cellular stress. And the extrinsic pathway is triggered by external signals such as ligand receptor interactions. But these pathways are going to involve a cascade of molecular events that lead to the overall apoptotic process. But cancer, going back to the importance here in cancer, there's defects and dysregulation in this apoptotic or apoptotic pathway, and it confers the resistance to cell death even longer. Chronic inflammation is persistent and prolonged inflammation, and this inflammation that can occur with infection, autoimmune disease, or prolonged exposures to irritants is an important factor in the development of the progression of cancer. Inflammatory cells, including the immune cells like the macrophages and the lymphocytes, they play a critical role in the inflammatory response. Remembering, the cells release cytokines, they signal molecules that regulate inflammation, and the immune response continues to unfold. However, with chronic activation of inflammatory cells, there's a sustained release of cytokines. This can promote the development of cancer. H. pylori, or Helicobacter pylori, this is a bacterium that colonizes in the stomach, right? It's a major risk factor for cancer, however. And chronic infection with H. pylori leads to persistent inflammation. And we just learned that with persistent inflammation, there is persistent release of cytokines or sustained release of cytokines. This is going to increase your risk for cancer. H. pylori is prominent in peptic ulcer disease, or PUD. Tumor-associated macrophages, or TAMs, they are a type of immune cell that infiltrate the tumor the microenvironment of that tumor. And they play a very significant role in tumor progression and survival. Associated macrophages, or TAMs, they are associated with the worst prognosis in cancer patients because they have a pro-tumor function. TAMs are key cells that promote tumor survival because they interact with cancer cells and then they undergo specific changes and function within the tumor. And this contributes to growth, invasion, more angiogenesis, and immune suppression. Unlike the typical function of a macrophage to phagocytose and eliminate harmful cells, TAMs, or tumor-associated macrophages, have a diminished cytotoxic response against tumor cells. So they become reprogrammed now to adopt an immunosuppressive phenotype. And this limits the ability to kill the cancer cells directly. So they block. TAMs develops the capacity to inhibit functions of the T cytotoxic cells. So remember, they can actually lyse or phagocytose, but they're going to inhibit that, and the natural killer cells. And these are critical components of the anti-tumor immune response. But they're secreting various factors and expressing cell surface molecules that now are impeding this activity of immune cells. Therefore, it's creating an immunosuppressive environment. And immunosuppressive environments favor tumor growth. And lastly, let's talk about the cytokine production in a little bit more detail. TAMs play a role in promoting tumor growth and spread because of the production of the cytokines, specifically interleukin 10 and transforming growth factor beta, TGF beta, which can have immunosuppressive effects and contribute to tumor immune evasion. Additionally, TAMs can produce factors that stimulate more angiogenesis to occur, and then therefore there's new blood vessels nourishing this growing tumor. In terms of the immune system in cancer, the immune system obviously plays a critical role in protecting the body. However, in immunosuppression that fosters the growth and the spread of cancer, this can lead to poorer outcomes. The immune system has mechanisms in place, right? To detect and eliminate the cancer cell, immune cells such as T cells and natural killer cells. They recognize and destroy cancer cells through direct killing, right? This is through the cell-mediated response. And they trigger this immune response against them, but the immune surveillance helps to prevent the formation and growth of tumors. This is the normal immune system protection. But immunosuppression, which is the weakening or impaired immune system, creates an environment that's conducive to development and progression of cancer. What can contribute to immunosuppression? Well, certain medical conditions, if you have chronic infection, certain medications can make you more immunosuppressed, such as glucocorticoids, or treatments even like chemo and radiation. There are specific examples here of normal immune systems protecting against cancer and what immunosuppression can foster. And you can see that with non-Hodgkin's lymphoma and Kaposi sarcoma, these are examples of cancers that are strongly associated with immunosuppression. So in individuals with compromised immune systems, like think about patients that have HIV or AIDS and they're undergoing organ transplantation, or the risk of developing these cancers are going to be significantly increased compared to the general population. And you can see that with non-Hodgkin's lymphoma, you are immunosuppressed 10 times more. And with Kaposi sarcoma, it's actually 1,000 times more. Understanding the interplay between the immune system and cancer has led to the development of immunotherapies. And this helps to restore the immune system and the immune response against cancers. Tumor markers or tumor cell markers, they're also known as biological markers. They're substances produced by the cancer cell, and they can be detected in bodily fluids like blood, CSF, or even urine. And these markers provide information that lead to understanding the prognosis or diagnosis, and even helping us to monitor whether a treatment is working or not. So we're monitoring that response. In terms of hormones, some cancers, such as breast or prostate cancer, they produce hormones or hormone-like substances that contribute to tumor growth and progression. And so we can use these hormone markers. Think of estrogen or prostate-specific antigen, or PSA, that we can obtain from a blood sample in a male. This can measure the progression. It can also help us just to monitor the trend. It doesn't always necessarily mean that there's cancer, but we keep an eye on it to see if whether we think that it warrants a biopsy, so that we can look at the actual tissue. So it helps us to monitor as well if there is a cancer diagnosis already there. Certain enzymes are produced by cancer cells, and they are released into the bloodstream. You think of lactate dehydrogenase, or LDH, or alkaline phosphatase, ALP. This can indicate a presence of cancer, and it helps to promote insight on the disease progression and responses to different treatments. Genetic alterations or mutations in the cancer cell can also be identified through gene markers. And these markers may include gene rearrangements, amplification, or mutations that are associated with a particular type of cancer. Tumor cells may express specific antigens on the surface, and they can be targeted by the immune system or used as markers as well to help diagnose. For instance, the carcinoembryonic antigen, or the CEA, we can use this for colorectal cancer, and the epidermal growth factor receptor 2, which we talked about, the HER2, for breast cancer. These are examples of antigens. And then for antibodies, produced by the immune system in response to cancer cells, they can also serve as a tumor cell marker. For example, if we have a presence of a specific antibody against an antigen, like human chorionic gonadotropin, or HCG, we can know that there's probably a presence of certain cancers, such as testicular or ovarian cancers, pregnant. So not diagnostic always, but it can give us insight that because the antibody is there, that there is a presence of these types of cancers. So what are the clinical applications? Tumor cell markers have important clinical applications because we can use them for early cancer screening, detection, monitoring, or even trying to understand the treatment response of our patient. We can also use to surveil or for surveillance of the patient's condition while they're going through treatment. There are some limitations though. Tumor cell markers don't provide everything. They're not specific to a particular type of cancer, and their levels can be influenced by other things than cancer. So just because we see an elevation, we want to look for trends and understand the full picture of the patient. We need to learn to screen and identify high-risk individuals. They can be utilized, but we need to recognize who really is at high risk, who's going to benefit from this further diagnostic test or preventative intervention. And does the literature say that we need to do this? What have we found? Because if it doesn't and we don't have support, then the insurance companies aren't going to pay for these biomarkers. It's important to note that tumor markers alone are not sufficient or definitive cancer diagnosis, and so they're just being used in conjunction with other aspects of clinical evaluation and diagnostic tools. Elevated levels of specific tumor markers can suggest the presence, but we still need to have further diagnostic procedures and know that they are not inclusive to a single type of cancer. Okay, so really important that we understand it's not diagnostic, but we can use it to gain more information and more insight. And we can continue to monitor the course of the cancer using this, but there are limitations to this. Also, there are a lot of false positives. There's elevated levels of tumor markers and non-cancerous conditions, and then also false positives. I'll give you an example. I had a patient with an elevated PSA, which typically would indicate a concern for prostate cancer. However, his elevation to do with some external factors. He was an avid bike rider, riding at least 50 miles every weekend. And over time, I saw an increase in trend of his PSA. Now, he was in his 60s, and so this is a time where your PSA tends to change, or you see prostatic changes. So I'm asking him about his ability to void and how this has changed, because maybe he has BPH, which is benign prostatic hyperplasia. But I'm getting his PSA, it's elevated. I tell him, come back in 3 months and let's check again, while knowing that he has been riding his bike a lot. And he said, okay, for 3 months, I will just not be as aggressive and see what happens. And of course, it went down. So all this to say is that your PSA, or prostate-specific antigen, is not as specific as we hope it to be, because it can be affected by outside or other internal factors as well. Now we're going on to part 2 of our cancer presentation, and we're going to think about it in terms of classification of cancer. Now, tumor classification systems are intended to help to stratify the risk for our patients, and the ability for us to understand this is helpful in the diagnostic and the prognosis and the treatment. The ability of malignant tumor cells to invade and metastasize is the major difference between a benign and malignant neoplasm or cancer. So in general, benign neoplasms are well differentiated, and malignant neoplasms range from well differentiated to under differentiated. Other differences between benign and malignant neoplasms we've already talked about in the first slide. So we use classification systems to provide a standardized way to communicate the status of a cancer to members of the healthcare team or families, to assist in determining the most effective treatment plans, to evaluate treatment plans, and to predict prognosis, and then again to compare in terms of groups for statistical purposes. That is why classification is important. So let's look at the 4 grades of abnormal cells. So first grade, this is when the cell differs slightly from the normal variation. So we have some mild dysplasia, and there may be well differentiated or low grade changes. In grade 2, cells are more abnormal, so we have moderate dysplasia and moderate differentiation. Grade 3, very abnormal, so we have severe dysplasia and poorly differentiated, so this is a high grade cancer. In grade 4, cells are immature and primitive, so we have anaplasia. They're undifferentiated, and the cell of origin is very difficult to determine. So this would be a very high grade. When someone refers to grade X, this means that they can't assess the grade. We also can talk about cancer in terms of clinical staging. So examples of diagnostic studies that may be performed to assess for the extent of disease, including radiological studies, so we can use bone and liver scans, ultrasound, CT, MRI, a PET scan. Well, surgical staging refers to the extent of the disease as determined by surgical excision, exploration, and lymph node sampling. So, for example, you have a laparoscopic approach to a splenectomy, taking out the spleen. This can be performed in staging of Hodgkin's lymphoma. So during the staging laparotomy, the lymph node biopsy can be done, and the margins of any masses can be marked with clips. They're usually metal clips, and they're used during radiation therapy. Then an exploratory surgical staging can also be used less frequently as non-invasive diagnostic technology becomes more readily available and sophisticated. Now, after the extent of the disease is determined, then we can say the stage has been, classified, and if the classification hasn't changed, the original description of the extent of the tumor remains part of the record. if additional treatment is needed or the treatment fails, we can do a retreatment, and then there would be retreatment staging done to determine the extent of the disease before they actually figure out what that retreatment is going to look like. So if there is restaging, they're going to use the R in front of the T and M, and this is going to help differentiate between the stage of diagnosis versus the restaging of the diagnosis. So we talked about cancer in situ, and now we're going to look at other, like stage 0 is cancer in situ. Stage 1 cancer means the tumor is actually limited to that tissue of origin and it's very localized. Still growing, but very localized. A stage 2 cancer means that there has been some spread, but it's still within that local area. Grade 3, there's been extensive local and regional spread. And then grade 4, we have metastasis. There's other malignant diseases like leukemia that we do not use the staging approach for. So look at this example of the TNM classification in terms of picture. TNM staging can't be applied to all malignancies, like I said, so leukemia being one, because it's not a solid tumor, and then therefore it can't be staged using these guidelines. Carcinoma in situ has its own designation in the system. We use the letter T, and then we'll put a small is subscript. Since it's not all the histological characteristics of cancers aren't there. So we can't use a TNM staging. But T stands for the tumor size and the invasiveness, and then N stands for how it's spread to the lymph node or not, and then M is for metastasis. So here you can see for tumor we have T1 through 4, nodes 0 through 2, and metastasis 0 through 1. And you can see the level of involvement that is described on the slide there. Okay. I have a quick case study that you can go through. HJ is a 30-year-old male who discovers a lump on his testicle and schedules an appointment with his primary care physician. And after testing, he's diagnosed with testicular cancer. I want you to think about how to explain the 3 stages of cancer and what metastasis actually means, because he's going to ask you and you want to know how to explain this to him. He's told by his doctor that he has a T2N1MO testicular cancer. So what does T2N1MO mean? So use the previous slide to help you to understand and to decide what this means. T2 means that cancer has spread to the blood or lymph vessels near the tumor. N1 means that it's spread to at least one lymph node, but there is not a lymph node that's larger than 2 centimeters. And M0 means that there is no metastasis at this time. Pathologic evaluation of a tissue sample is the only definitive means to diagnose cancer. A pathologist has to examine the tissue and determine if it's benign or malignant. After that, they're going to determine the anatomical tissue, where the tissue arises, so the histology, and then the degree of cell differentiation. So they're going to provide a histologic grade. Other information that can be obtained includes the extent of malignant involvement, so like what is the size of the tumor and the depth. And then the evidence of invasiveness, so was it extracapsular, lymphatic, adequacy of surgical excision, is it positive or negative for surgical margins, nuclear grade. Again, when we're talking about nuclear grade, we're talking about that mitotic rate that was on the first slide. And specific staining techniques can also provide insight on how responsive the tumor might be to a specific treatment. So we can actually also get receptor status and tumor markers as well.